Thanks for joining us this morning. I'm Dr. Jamie Wilbur. I'm the Michigan State University potato and sugar beet pathologist. My program focuses on integrated pest management in for potato diseases. Currently on this research farm, we do have foliar soil-borne disease trials out in the field. We have some white mold work with some interesting new weather stations and looking at disease predictive modeling. But today, we're actually focusing on the research of one of my wonderful graduate students. Emma Schlachter is here with us. Emma, if you want to wave your hand. She is a master's student in our program. She's had the pleasure of going through the COVID-19 epidemic of last year and trying to continue her research, aiming to finish up in 2022. So she's been an excellent researcher in our program. We've been very privileged to have her with us. Her project is looking at post-harvest diseases, looking at surveying and characterizing diseases that we have here in Michigan storages, but also management of those diseases in collaborative research projects. We're grateful to have the collaborations that we have with Chris Long's program, the Potato Outreach Program, as well as the Potato Breeding Program. Some of those collaborations are directly connected to this work today. Over the last several years, our program began in 2018. Since then, we've been looking at the different diseases in Michigan, and we've identified some of the major rotting diseases impacting our potatoes in storage, two of which I'm showing on this poster here. It's a little bit bright today. I hope you can see this. So can anybody tell me what this disease is here in this upper right-hand corner? My right hand. Anything? Any guesses? What was that? Fusarium. Fusarium, yes. Fusarium dry rot. Thank you. So Fusarium dry rot is one of our top diseases that we generally detect in samples in both seed and commercial storages. And so this rot is characterized by a dryness. So other rots of potatoes are smelly, they're wet, they collapse, they leak. This one is dry, and it doesn't have too much of a scent in and of itself. From the outside, we can see that these potatoes look fairly clean, fairly normal from the outside. When we cut them open, that's when we get the Easter egg effect of finding the treasures inside, for a pathologist anyway. So this is a kind of a white, crumbly rot inside the potato tuber, and this can dry out and form cavities inside of the tuber. As Celeste was showing us, different species of Fusarium have different colors, so sometimes we do get colorful fungal mycelium, fungal tissue inside of these tubers. The second disease that I'm showing today is this one here in the center panel. Can anybody have any ideas about this particular disease? The name is somewhat descriptive. That's the hint.
Pink rot.
Pink rot, excellent, thank you. So pink rot is showing here in the center. This disease is characterized by its pink coloring upon cutting. And so after you've cut, once you cut, it might look somewhat normal. 10 to 20, 30 minutes later, you usually get this pink coloring of the tissue. Otherwise, the tissue is somewhat spongy, so it doesn't cause a complete tuber collapse. The other two diseases that my program works with is a pythium leak. That can cause kind of a tuber collapse over time. It leaks out the cellular contents. And these diseases also open up the potato tuber to other secondary breakdown. And so the last one that we work with is bacterial soft rot. Thank you, Emma. So bacterial soft rot, of course, opens up the potato tuber to that cottage cheesy rot. That brings in a lot of secondary bacteria, which cause the smell that you may have associated with rotting potatoes in storage. Our research objectives, we had two in Emma's work. So the first one was looking at variety evaluations against two of these diseases, Fusarium dry rot and pink rot. We had 38 entries in her research of different standards, and I'm showing the standard varieties in bold black here, and an additional advanced breeding lines through collaborations with the breeding program. Excitingly, we do have breeding lines that are performing well against both Fusarium and pink rot in our evaluations. These were after 7 weeks of incubation after inoculation. And so the varieties in blue are our best performing lines and varieties. They're exhibiting only 10 to 25% of the symptoms that we observed in our worst performing entries. The pink lines obviously are statistically similar to our worst performing entries. So we do have a range. I'm showing an abbreviated summary, and that's also available on your handout today. One note here, in our grouping column, anything with the same letter is significantly similar to each other. And these letters in parentheses are different locations. So we can see some interesting differences from that originate from agronomic management practices that differ between different commercially growing locations or research locations that have caused these different material to perform differently against these storage pathogens. For example, the Petoskey, there at the bottom of the Fusarium dry rot table, from one location it performed very well. In another location, that material did not perform well against our Fusarium. Similar in the pink rot, different locations, it performed moderately to very well. But this is something that's all going into the information of our breeding lines and our breeding programs. We have 3 breeding lines that have been identified in our screen, and they are currently being used as parents in the breeding program. So we're excited to have some of this resistance. We're same amount of time, but we're getting significantly less symptoms in some of our breeding lines that we could have. So we're excited to see that going forward. The last objective, we had a storage here in our demonstration storages. We actually had inoculated potatoes. We put in 2 different bins. One bin was treated with a Sanidate 5.0 application. That's a fogging application. There was some interest of whether this had impact on disease progression. In our research, showing on the bottom, this is our first year testing this product, but we did not see a significant difference between the Fusarium dry rot or the pink rot development in our treated and non-treated tubers. And in the bottom center of this is a Fusarium progression in a treated bin and in our control bin. And that was after 7 months in our commercial storage, or research storage under commercial conditions. So we're excited to test this product again, and we're happy to take any questions as we're moving forward.
What's the white?
The white here, these are standard varieties. The white is that they performed moderately, so not the best and not the worst. But I just wanted to include the standards on here for comparison.
We used to get some control with Myrtec on Fusarium in Michigan. Is everything, all the Fusarium we have now, is that resistant to?
That's a good question. There is no resistance to Myrtec. I'd have to look at some of the different species. So we have several different species of Fusarium that we've identified in our surveys. We're continuing to characterize those. Once we know more about the species population, we would know a little bit more about historically how much resistance they've had to Myrtec. Thank you for that.
Are the tubers pretty well infected before they're getting into storage then?
Actually not. We inoculate them about a couple days before we load the bin. So we wanted it to be infection under those commercial conditions. So inoculated, they sit for a day and then they're put in. And these are bins just like the ones behind me. We load them from the bottom up and we layer our inoculated tuber sample bags at 4 layers throughout the entire pile. So we are getting a represented replication.